So let us discuss bacterial cells. Now, if you haven't already looked at the tutorial I have done that looked a little bit like this, well, exactly like this, on plant and animal cells, I do encourage you to have a look at that, because we're going to make an assumption that you guys understand the difference between eukaryotes, eukaryotic cells, eukaryotes, and prokaryotes. Okay, so I'm going to make that assumption because straight away, I'm going to start talking now about these prokaryotes, because bacterial cells themselves and archaea are what we call prokaryotic cells. Prokaryotic cells, okay? Prokaryotic cells. So we're interested in that. Now, again, just let me repeat what I said. We are talking about bacteria. Obviously, the title of this tutorial tells you that, but we are also talking about a structure called archaea. AEA on the end of that. AEA on the end of that. Now, archaea is a really fascinating biological structure, a structure that which was, if not unknown, was kind of unrecognized as a living organism for a long period of time, and is a great source of study. Who knows, maybe you will be someone that goes on to study and investigate and to solve some of the mysteries around those kinds of structures. But all that being said, we want to understand a little bit more of these prokaryotic structures. Now, little bit like last time, I'm going to start this off by drawing what is rather ugly, but still it's a shape of what we might call a cell membrane. Okay, so we have here a cell membrane, a little bit like I had with the big picture of the plant and the animal cells earlier on. One of the big differences here, however, is that our DNA, which I've drawn almost accidentally like some kind of staggering dinosaur stroke raccoon shape here, but our DNA in this case is, normally with our other cells we would be drawing a membrane around that, but in this case we have no membrane. So the point we'd say here is that we have our DNA. Sorry, let me go back to the right color. We have our DNA. Okay, but our DNA is not membrane bound. Okay, not membrane bound. Okay, so it kind of is in the cytoplasm. Okay, it's in the cytoplasm. Now, again, we can kind of, we can kind of sort of sketch that cytoplasm in here. It's that kind of fluid-like structure. But notice here, there's no separation of that fluid with the DNA itself. So we can also add the point here that our so at least in the prokaryotic structures, our genetic material is not enclosed in a nucleus. Now, sometimes, by the way, we call it a nucleoid in this context, okay, a nucleoid, but more of that in another case. But the other point I'd like to make about this DNA and our prokaryotic structures is we also need to understand it's what we call, or what we often describe it as, a single DNA loop. Okay, I've quite specifically drawn this DNA in a single loop. If you compare that, albeit to what is an ugly drawing of this sort of scraggy sketch of sort of jumbled lines over here, we'll see that this is kind of much more ordered, or at least it's a single loop. We sometimes call them single rings. Okay, single rings. Now, this is a generalization, understand, but we might call them single rings of DNA, and that involves a structure called a plasmid. Okay, a plasmid. Again, more of which once we get past this kind of big picture stage. My last point to you guys is I want to raise an interesting point with you. When we talk about our prokaryotes, what we're talking about now, of course, we tend to find that they are much smaller than eukaryotic cells, than eukaryotes. Okay, much smaller than eukaryotes. Okay, so this cell, although I've kind of drawn it approximately the same scale, in reality we might find with this eukaryotic structure that it's significantly smaller. Okay, significantly smaller. We tend to find that a plant cell, for example, will be between 10 and 100 micrometers. but an animal cell will only be up to about 30 micrometers potentially. Okay, so it can be sort of up to 3 times smaller, or a little more than 3 times smaller. So we're talking about a significantly smaller structure than what we did in that previous session, which was our plant and animal cell.